In this section, we are going to discuss the internal structure of dicot and monocot leaf. First, we will see the structure of the dicot leaf, that is, the transverse section of a dicot leaf. Now, this is the leaf, dicot leaf, where you have midrib, veins, and veinlets. Now, if I am taking a cross section of this leaf, I will see the section enlarged in the microscope. Let us study the internal structure of dicot leaf. The example given in the textbook is mango. Now, dicot leaf consists of two epidermis, upper epidermis and lower epidermis. This is upper epidermis. And this one is the lower epidermis. First, we will see the upper epidermis. It is the outermost layer of the leaf. It is single layer made up of only parenchymatous cell without any intercellular spaces. Of course, stomata is found, but they are less in number, distributed here and there. You have one more layer above this epidermis that is called as cuticle. Cuticle is made up of waxy substances called cutin, which prevent the excess loss of water. Now, the lower epidermis. Lower epidermis is also made up of single layer of parenchymatous cell, but here it is interrupted here and there by many stomata. The distribution of stomata in the lower epidermis is more when compared to the upper epidermis. But you also have one more layer below the upper epidermis. This is called as cuticle, but the cuticle is thin in the lower epidermis than the upper epidermis. Now, since the stomata is more in number, exchange of gases and transpiration, that is loss of water, is facilitated by the lower epidermis. You're also able to see the epidermal outgrowth, that is epidermis hair is also found in the lower epidermis. Now, the tissue that is formed in between the upper epidermis and the lower epidermis is called as mesophyll tissue. So, from this layer to this layer, it is called as mesophyll tissue. I am writing this side. This is called as mesophyll tissue. The tissue that is found between the upper epidermis and the lower epidermis. In fact, the entire tissue that is found in between the upper epidermis and the lower epidermis is called as mesophyll tissue. The mesophyll tissue in dicot leaf is differentiated into palisade parenchyma and spongy parenchyma. Now, this is the structure or the layer that is formed below the upper epidermis constitute this palisade parenchyma. So, this is palisade parenchyma. Palisade parenchyma is formed below the upper epidermis. They are elongated cylindrical cells without any intercellular spaces. They are usually one layer or two layers. When you see they have more number of chloroplasts, so their function is photosynthesis. Now, this below this palisade parenchyma is the spongy parenchyma. So, this is spongy parenchyma. Now, spongy parenchyma doesn't have a proper shape. They are irregularly arranged with lot of intercellular spaces. When you see the number of chloroplasts, chloroplast is of course found in the spongy parenchyma, but they are less in number. Since they have more intercellular spaces, their function is exchange of gases. So I repeat, mesophyll tissue is nothing but the entire tissue that is formed between the upper epidermis and the lower epidermis is called as mesophyll tissue. In dicot leaf, mesophyll tissue is clearly differentiated into palisade parenchyma and spongy parenchyma. Whenever you see a clear differentiation of palisade parenchyma and spongy parenchyma, such leaf should be called as dorsiventral leaf. In fact, all the dicot leaf is dorsiventral. That means there is a clear differentiation of palisade parenchyma and spongy parenchyma. The third one is the vascular bundles. The vascular bundle is prominently found in the midribs. So this is the midrib. Here is the place where the vascular bundle is found. And moreover, the vascular bundle in the midrib will be bigger than in the veins. The vascular bundle in the dicot leaf is conjoined, collateral and closed. The vascular bundle in dicot leaf is conjoined, collateral and closed. We have already studied all these terms in the previous classes, but let me explain this once again. What is the meaning for conjoint? When the xylem and the phloem are arranged in the same radius, then it is called as conjoint vascular bundle. Now what is collateral? Xylem and phloem are arranged in the same radius with the phloem towards the periphery is called as collateral. Now what is the meaning for closed vascular bundle? The xylem and the phloem is not separated by cambium, so it is called as closed vascular bundle. So in dicot leaf, the vascular bundle is conjoined, collateral and closed. Now in dicot leaf, the vascular bundle is surrounded by a compact layer of cells made up of parenchymatous cells. This is called as bundle sheath or border parenchyma. Now the xylem and the phloem is surrounded by a compact mass of parenchymatous cell called as bundle sheath or border parenchyma. So bundle sheath, this layer is called as bundle sheath or otherwise called as border parenchyma. Now when you see the arrangement of the xylem and phloem, xylem is facing towards the upper epidermis and phloem will be facing towards the lower epidermis. In fact, the protoxylem, the first formed xylem is facing towards the upper epidermis and the metaxylem is facing towards the lower epidermis and phloem is also facing towards the lower epidermis. For that, you have to clearly understand, or you can simply write the xylem is towards the upper epidermis and phloem is towards the lower epidermis. So this red color region, this is called as xylem and this one is the phloem. So that's all about the internal structure of the dicot leaf. Let us move on to the internal structure of a monocot leaf. The example given in the textbook is grass. Monocot leaf, you are able to see two epidermis. This is upper epidermis and this one is the lower epidermis. In monocot leaf, there is no such differentiation as we saw in the dicot leaf. In the dicot leaf, we saw the cuticle is thick in the upper epidermis, whereas the cuticle is thin in the lower epidermis. The distribution of stomata is less in the upper epidermis, but it is more in the lower epidermis. But in monocot leaf, there is no such differentiation. You have a single layer of parenchymatous cell without intercellular spaces. Of course, stomata is found, but it is equally distributed in the upper epidermis and the lower epidermis, and the cuticle is also moderately thick both in the upper epidermis and the lower epidermis. Sometimes you will be able to see on the upper epidermis thin elongated cells will be seen both in the upper epidermis as well as in the lower epidermis, but in upper epidermis it is very prominent. So these cells are called as bulliform cells. So these cells are called as bulliform cells. So what are called as bulliform cells? Sometimes the upper epidermal cells will become thin and elongated, and these are called as bulliform cells, which is a characteristic feature of all monocot leaf. The function of bulliform cell is rolling and unrolling of the leaf in the monocot leaf. Now the tissue that is found between the upper epidermis and the lower epidermis is called as mesophyll tissue. So I am writing this, the tissue that is found between the upper epidermis and the lower epidermis, all the entire tissue is called as mesophyll tissue. Now in monocot leaf, the mesophyll tissue is not differentiated into palisade parenchyma and spongy parenchyma as in the dicot leaf. So all monocot leaf is called as isobilateral leaf. So all monocot leaf is called as isobilateral leaf. That means there is no clear differentiation of palisade parenchyma and spongy parenchyma, whereas in dicot leaf there is a clear differentiation of palisade parenchyma and spongy parenchyma, so it is called as dorsiventral leaf. When you see the mesophyll tissue in the monocot leaf, you will be able to see same elongated cells arranged, homogeneous layer of cells arranged with intercellular spaces, and they have got chloroplast. Next is the vascular bundle. Now in monocot leaf, the vascular bundle is arranged in a different manner. That means you'll be able to see a bigger vascular bundle followed by a smaller vascular bundle, again a bigger vascular bundle and smaller vascular bundle, and so on. Now on every bigger vascular bundle, on the either side of the bigger vascular bundle, this upper side and the lower side, you'll be able to see a patch of cells made up of sclerenchyma. And these sclerenchymatous patches are found only in the bigger vascular bundle and not found in the smaller vascular bundle. On the smaller vascular bundle on the either side, you will not be able to see the sclerenchymatous patch. Now this vascular bundle is conjoined, collateral and closed. You know already the meaning for conjoined, collateral and closed. And every vascular bundle is surrounded by a thick layer of parenchymatous cell called as bundle sheath. So here the cells of this bundle sheath are a little bit bigger in size. Now the vascular bundle consists of xylem towards the upper epidermis and phloem towards the lower epidermis. Now what is the main difference between dicot leaf and the monocot leaf? All dicot leaf is dorsiventral and all monocot leaf is isobilateral. You know what is the meaning of dorsiventral leaf? When there is a clear differentiation of mesophyll, that is mesophyll is differentiated into palisade parenchyma and spongy parenchyma, you call this is dorsiventral leaf. When there is no such differentiation, when the mesophyll is not differentiated into palisade parenchyma and spongy parenchyma, then it is called as isobilateral leaf. That's all students. We have studied the internal structure of dicot and the monocot leaf. Please go through your textbook for the diagram and all the notes. Thank you.